Of novel anti-cancer compounds targeting mitochondrial protein homeostasis, which will be delivered by Professor Dr. Waleed Houri, who is a professor at the Department of Biochemistry at the University of Toronto. Thank you very much, sir, for coming. We are honored to have you here with us today. So I would like to formally begin the session by the recitation of a few verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Jazakallah. Now I would like to invite Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech, for his opening remarks.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. My dear Professor Dr. Waleed Houri. Waleed looks so familiar that, when I just met him, I said, I probably met you hundreds of times. And he reminded me that he actually visited Karachi. So you can see aging is taking the toll. But the possibility that I come across Waleed when I was in Cornell. I was in Cornell between 1987 to 1994. And at that time, I think you were doing your PhD. I was in the laboratory of Professor John Clardy. And that's right. Yeah, and then I used to work with Professor Weinrup. So long memories of Ithaca. We used to live very close to Hanging Bridge and then Hasbrook. I'm sure that's somewhere close by, or maybe at the prayer place, because we all used to pray together. And initially there was a small place in the church, and later there was a house which was purchased in 1988 or 1989. I think I learned that later a masjid was built. Yeah, maybe later after that. So we used to go, and I still remember that early in the Fajr, when temperature was below 7 or 8, we used to go for Fajr, and then a police car used to follow, and they used to see so many boots outside the house. Because we used to remove our boots and to go into the masjid, and they were surprised why people just remove boots outside and what they do in the morning, early in the morning, before the sunset. So even at that time, we used to have lots of experiences, and Ithaca was a beautiful place. So nostalgia of all kinds. Waleed is a professor of biochemistry in the Department of Chemistry at the University of Toronto, which is one of the most prestigious institutions, it's among the top 50. Dr. Waleed obtained his MSc in 1991 and PhD in 1996 in Cornell University. And then did his postdoctoral training at the Sloan Kettering Institute in New York City and at the Max Planck. Then he moved to Germany, again to a very prestigious institution of Max Planck, biochemistry in Munich. And then he has been, engaged in very high quality research. He is interested in general area of cellular stress, responses and role of molecular chaperones, ATP-dependent proteases, and these responses. So it's a very intricate biochemical balance which is related to protein repairs and many proteopathic diseases. Dr. He is also heavily involved in general scientific community in several capacities. His research interests, as I mentioned, primarily divided into three main areas. One is on R2TP chaperones complexes. The second is on the CLP system, and third is on mapping the chaperone interaction network. The three projects deals with the common theme of cellular stress responses. He is also interested in identifying compounds that target protein cellular homeostasis, and that can be developed as anti-cancer and antibacterial compounds. His research endeavors are aimed at understanding the role of molecular chaperones and ATP-dependent proteases in modulating protein homeostasis and under different stress conditions. There are two main projects that he has been currently pursuing. One is in the role of HSP100 CLP system in cellular stress, and the other on HSP90 R2 TP system. So he has been engaged in hardcore biochemistry of cellular stresses. More importantly, he is very passionate of delivering his knowledge to the young people. And his presence here after a very tiring journey from all the way from Toronto, 13 hours flight to Islamabad is a proof that he is so passionate to be connected with the Muslim community, not only in Pakistan, but through Comstech to the OIC. And we are so honored to have him. Waleed, thank you very much for coming. You remain young, as I saw you years ago, and I'm sure that you would remain young with your commitment, your perseverance, your motivation, and your energy. Bismillah ar-Rahman ar-Rahim. Please go. Thank you.
So thank you very much, Professor Iqbal Choudhury. This has been really a pleasure to be here. And definitely I have a very fond memories of Cornell where we first met. It was one of the foundations of our friendship. And I'm glad that I could see you again after such a long time. And you look as young as you were before. So thanks again for organizing this visit for me to come to Comstech. And it has been really a very nice event for me. It has been a long flight, definitely, but something I really enjoyed. And thank you again for the invitation. So I look forward to learn more about Comstech through my interactions with you and your colleagues, and how Comstech is trying to promote science in this region, in the OIC countries. I also want to thank actually the staff of Comstech, especially Ms. Khazima and Ms. Maham, for organizing my visit. And just yesterday, we had the chance to go and see several landmarks in Islamabad, including the Faisal Mosque and Lok Versa. And I was there with the Indonesian delegation. So it was very interesting interaction. And I learned a lot about the history and the struggles of this place and how this great country came about. and the development, both scientific, cultural, and economic, that Pakistan has went through. And it's quite amazing to see. So thank you again for the opportunity. So for my presentation, I will be talking about my research, one aspect of my research, which combines hardcore biochemistry with hardcore chemistry, as well as structural biology, whereby we are trying to identify new compounds with new mode of action that can be developed as anti-cancer compounds. So if I can have my presentation, yeah. Okay, so thanks again. So this effort I will describe to you today is basically the hard work of several members of my group. And I'd like to acknowledge at the beginning, Dr. Mark Mabangalo, who's started this effort. And then there's Dr. Keith Wong, Mariam, Elisa, Funing, and Emily, who have been all part of this effort. And we've had many collaborations. So this is one thing that in my lab we do. We actually collaborate heavily with multiple groups to try to push the project forward, especially Robert Beatty, who's helped us with the medicinal chemistry, Anne-Claude Dungeras, who's helped us with the mass spectrometry, Louis K. with NMR, and we've collaborated with a company, which I will discuss later, with Ed and Lee, and finally Aaron Schimmer at Princess Margaret Cancer Center. So now I'm gonna go directly into the science, and oops, lost the presentation. Oh, it's still there, okay, 1 second. This one here?
Yeah.
Okay, yeah. Okay, perfect. So if we take a look at something like the mitochondria, and we look at protein homeostasis inside the mitochondria, so there are molecular chaperones as well as ATP-dependent proteases that maintain protein homeostasis. And what we mean by that is that there are these quality control proteins that ensure that whatever proteins are in the mitochondria are properly folded, properly assembled, and if need be, properly removed, properly degraded. So I'm showing you here an example of some of the chaperones, so like HSP60 and HSP10. So this one. So HSP60 and HSP10. So this is the chaperone family that ensures proper protein folding. Oh, we lost it. So let me. Okay, now it's advancing. Okay, I hope everyone can see it online and here. So technology is always amazing and sometimes causes us problems. But okay, so what I was describing to you here is some of the chaperones that are involved in ensuring proper folding of proteins like the HSP60 and the HSP10. And this is shown here. But there's also protein degradation systems that degrade these proteins if need be. And the major one that I want to discuss with you today is the ClipXP complex. And that complex is this one here. So this complex takes a folded protein, unfold it, and degrade it into small peptides. So if you look at how this complex looks like, so the ClipP is a protease, so that's where the P comes from. So it's a proteolytic complex consisting of 14 different subunits. And each subunit, you can think of it as having the structure whereby you have the handle region as well as the head domain. So the handle region is where the helix and the strand are. So this is helix E and the strand 9. And these helices and strands come together and form the complex. And the complex is a tetradecamer, meaning it's a 14 subunit complex. And if you look at the complex carefully, you can think of it as being 2 heptameric rings. So one heptameric ring, another heptameric ring, and they come together to form this complex. And what is important here is that proteins will be pushed into this ring and into the cylinder. So the CLIP-P is a cylindrical structure, and degradation happens inside that cylinder. It is a serine protease consisting of 14 subunits. So you have Serous asp catalytic triad. So it's a classical enzyme that cleaves proteins. And you have 14 of these active sites inside the catalytic chamber. And basically, it's a very processive protease because you can take a protein, push it into the cylinder, and degradation takes place inside this chamber. So in the mitochondria, the targets for this complex are established, but not very well understood or very well studied. So we know that this complex targets certain proteins in the mitochondria, and these proteins are involved in different pathways, like the Krebs cycle, or the pyruvate metabolism, or amino acid metabolism. And so the CLIP-XP complex is actually going after many of these proteins inside the mitochondria to ensure that they are at a certain level and that they are not overexpressed, and they are, if need be, that they are degraded when the time comes. Clinically speaking, why is this complex important? So if you look at the levels of the CLIP-P in certain cancers, we find that the CLIP-P is very highly expressed in certain cancers. And here I'm showing you an example of colon, liver, prostate, and uterus cancer. And actually, maybe it's easier if I can use this laser pointer. Okay, can you guys see the laser pointer? So it's easier for me rather than turning back. So you can nicely see that we have increased levels. So this is immunohistochemistry quantification. There's increased levels of the CLIP-P in these different cancers. And if you look at the Western blot here, you can see that the levels of CLIP-P are low. So this is a prostate tissue in the normal prostate tissue, but in the tumor part of it, the CLIP-P levels are quite high. So this verifies the targeting of CLIP-P to develop new anti-cancers. So how does the complex look like? So this is CLIP-X, is actually a hexameric complex, and the CLIP-P is this one. It's a tetradecamer that I mentioned to you before. The hexameric CLIP-X will dock onto the CLIP-P, and the CLIP-X is actually an ATPase. So it will take a protein unfolded, thread it through, and into the CLIP-P chamber where degradation takes place. So a protein will be degraded into very small peptides of around 10 residues. These peptides will subsequently be released, and then the cycle can repeat. So schematically, I can show you this cycle. Oops. Okay, it's slowing down. I'm not sure what happened. Is it the Zoom?
Presentation, I think.
Working on? So let me. Full screen. Okay. So I'll use the pointer again. Okay, yeah. Okay, so this is again a schematic of the ClipXP cycle whereby you have a folded protein. It's recognized by the cap, by the ClipX. It's physically unfolded in a ATP-dependent manner. So it's actually quite interesting because you have a protein physically unfolding another protein. So a protein acting on another protein to actually unfold it. So this is quite interesting by itself. And then the protein is pushed into the cylinder. So this is the ClipP cylinder where degradation takes place. It's degraded into very small peptides, and then the cycle can repeat. So this is the functional cycle of the ClipXP complex. So what I want to discuss with you are how we targeted this complex to develop new anti-cancers. And I will discuss the activation of the ClipP using compounds called ADAPs or other compounds called TR. And then I will discuss the consequences of the ClipP activation. So now you can ask me a question, is basically, ClipP is a serine protease. So why do you want to not just inhibit this enzyme, right? It's a serine protease, so you can easily inhibit it. And the problem with that is that we tried very hard to inhibit the ClipP, but we could not find specific inhibitors for this enzyme because any inhibitor we discover targets other enzymes in the cell, and therefore we don't have that specificity. So we went after another approach, which is activation rather than inhibition. And I will tell you what activation means. So I'll describe first these Acidpsy peptides, ADAPs. So the idea is very simple. How do we activate ClipP? So if you look at the complex itself, so this is the cylindrical structure of the ClipP that I described to you before. And if you look at the top, so basically you're looking at the cylinder from the top, you'll see that there are these hydrophobic pockets in purple. So there's 7 of them because it has a 7-fold symmetry. So you have 7 of these hydrophobic pockets, and this is where the ClipX docks. So what we found is that if we can find compounds that actually bind at these pockets, what happens is that we're going to open up that entrance pore. So the entrance pore opens up, and what happens next is that proteins will go unspecifically inside that entrance pore and get degraded. So we lost the specificity. So initially, we had a cap that told us what should be degraded. But now anything that can get into that cylinder will be degraded. And therefore, there's no more specificity to this complex. And what you have a protease that's chewing everything up inside the cell, and this leads to cell death. So I hope this idea is very clear. So basically, we're activating, or more specifically, we are dysregulating the protease. We're allowing things to be degraded unspecifically, and this leads to cell death. So this is shown schematically here, whereby you have a protein that's going to fall into that cylinder because then now we have an open pore, and then this protein gets degraded. So early on, we discovered these Acidpsy peptides. So these are these compounds that do that trick, that can activate the ClipP. And if you look carefully, this is basically a cyclical peptide, and that's why they are called Acidpsy peptides. They are more specifically Depsy peptides, if you're into the chemistry. So these are the ADAPs. Initially, they were described by a group at Bayer as well as by our group. And we initially as antibiotics, but subsequently we showed that we can use them as anti-cancers. And then I will describe to you these types of compounds, which we call the TR compounds, that do the same trick. So let me show you the experiments first. So we generated many analogs of these Acidpsy peptides, and we checked how good they can activate the ClipP. And this is a quantitative measure. RD25 means at 25 micromolar, we're adding these compounds at 25 micromolar and see how well the ClipP gets activated. The closer the number is to 1, this means the better is the activation. So as you can see, we have several compounds that can nicely activate the human ClipP protein. So subsequently, this is at 25 micromolar. We checked at 1 micromolar, so we're reducing the concentration. Several of these compounds fell off, and we have another set of compounds that are still active. And I'm just showing you the structures of a couple of these analogs. So to show you that the compounds do indeed work, so this is on a gel. You can run a gel. In this case, what we have is a model protein, let's say casein. It can be any other model protein, but casein is being used here. You have the ClipP, you have the ClipX, and creatine kinase is just an ATP regenerating system. So basically, if we have just DMSO, you can see that casein is not being degraded as a function of time. So there's no loss of that band. However, if we add our compound, then at low concentrations, we don't see much of an effect. But at higher concentrations, at 22 micromolar, we can see that the casein gets degraded as a function of time. So at the 0 time point, you can see the band, and then subsequently the band is lost. So this tells us that, yes, the ClipP is doing that trick. We are actually degrading a protein. And we did a very careful titration to get an idea of the stoichiometry. And basically what we see as we titrate in the compound, we see inhibition at the beginning. So the compound, as I told you, there's 7 docking sites and on the one ring, actually. So on a cylinder, there's 14 docking sites. And what we see is that we need at least more than 1 compound to bind to start the activation process. So we need at least 1 site to be occupied for activation to start to happen. And we did the titration to get the binding constant for these ADAPs, and we get a binding constant of around 0.5 micromolar. So this is how good these compounds are. They are in the 0.5 micromolar range. Or if you are actually a medicinal chemist, you'll say, okay, this is not very good. You should be in the nanomolar range, right? So currently we are in the micromolar range for these compounds. So when we first found these compounds, I thought these compounds are quite big. So I told my guys that I don't think these compounds would work on cells, because if you want to think about it, you have the compound, it has to cross the cell membrane and actually cross the mitochondria and enter the mitochondria to activate the ClipP in the mitochondria. So I did not think that these compounds would work. So I was telling Keith, my postdoc, saying, okay, don't do the experiment on cells. It's not worth it yet. We'll design a new set of compounds. But of course, my postdoc did not listen to me and went ahead and did the cell-based experiment. But he did it much more nicely than I was thinking of. What he did is the following. He used, here I'm just showing an example of the HEC293 cells. You can use any other cell type. And he added the compound, and you can very nicely see a titration of the ADAP, and you have an IC50 of around 0.5 micromolar. To show that these compounds are actually on target, that they are hitting the ClipP in the mitochondria, what he did is he used CRISPR to knock out the ClipP from the genome. And the compounds need the ClipP to be active, right? If there is no ClipP, there's no target, and therefore the compounds would not work. And this is what we see. So in the open triangles, you'll see that the compounds do not work until we go to very high concentrations, and then it becomes unspecific, binding to unspecifically to something else. But basically, in the absence of ClipP, the compounds don't work. In the presence of the ClipP, we have a very nice titration, and therefore the compounds are on target in these cells. So this is very important information that we gained. Okay, I think it's stuck again. Sorry. Okay, now it moved. Okay. So since we have titrated the, since we have many different analogs of these ADAPs, we checked how well the binding constant in vivo compares to the IC50 on cells. And we have a very nice correlation between the binding constant on the protein versus the IC50 on cells. So basically telling us again that these compounds are hitting the ClipP in the mitochondria. So next slide. It's not moving. Click, sorry. Just click here and then Okay, so let me just go back then. Okay. Okay, so then we checked these compounds against different cell lines, and we get IC50s in the 0.5 to 1 micromolar against different cell lines. And this is the levels of the ClpP that we see in these different cell lines. So sometimes the ClpP is low, other times we see that the ClpP is high, but nevertheless, these compounds are still acting on target. So what we found as well is that these compounds cause apoptosis. So this is shown here, whereby we see that when we add these compounds, we start seeing blebbing as well as condensation of the DNA as quantified by the tunnel assay. So there's DNA fragmentation. And this is the quantification whereby we have in the wild type cells a certain amount of DNA strand breaks. This is in the black bar here. While in the ClpP knockout, we don't have any effect. And this is very nicely shown here as well. So okay, now it's frozen again. Okay, so the other thing is that we see is that we have apoptosis happening because both the caspase 9 and the caspase 3 are being cleaved. And as you might know, the caspases, caspase 9 and caspase 3 are the intrinsic caspases. Basically, they respond to an intrinsic signal, and they are being cleaved to induce apoptosis. While the caspase 8, which responds to an extrinsic signal, is not being cleaved. So telling us that we have an intrinsic activation of the caspase-dependent pathway. We also can look at what's happening at the mitochondria. So this is shown here, whereby we have wild-type cells. If we treat them with the MSO for 24 or 72 hours, you can see in red the very nice mitochondrial network, and in blue is the nucleus. However, if you have the ADAP41 added at 24 and then 72 hours, you can clearly see that there's fragmentation of the mitochondria, indicating that there's mitochondrial stress and the mitochondria is basically facing problems. Another thing that we did was to look at the Aux Force. Okay. So now it's okay. This is not. It's going to be a very long presentation here.
सर प्रेजेंटेशन की हैवी है और एक्स्ट्रा वो यूज कर रहे हैं ना इसमें फीचर्स है actually the presentation is so heavy so
Okay, because of the memory on it. Yeah, I have a lot of structures, so that's why it's might be taking up a lot of memory. So we did Aux Force analysis on the compounds as well, wild type versus knockout. And you can just concentrate on these bars here. We can see that the wild type with the MSO, you have a certain level of basal respiration. However, if you have wild type with the compound in green, would be the levels of respiration have been reduced. The ATP synthase activity is the same thing. We have reduction in that in the wild type, proton leak, as well as non-mitochondrial oxygen consumption. However, if you do the same experiment with the ClpP knockout, so this is in red with the MSO and in purple with the compound, there is no difference. So basically, we have no effect on any cells that have no ClpP, again, telling us that we are on target. Finally, we also solved the crystal structure of the ClpP with our compound. And this is the structure that was solved by Mark in my lab. And as you can see, the compound is sitting where I told you before at these hydrophobic pockets. So this is 1, 2, 3, 4, 5, 6, and 7. So it's at the interface between two different subunits. And this is where we expected it to bind. So basically the compound binds at these hydrophobic pockets, prevents the cap from binding, and at the same time, opening up that entrance pore. So you can see this entrance pore very nicely larger compared to the apostate, where the entrance pore is much more smaller. So this is a zoom in into the interface where the compound sits. So you can see it sits at the interface of these two subunits, one in purple, the other one is in blue. And actually, if you look carefully, you can see that these two subunits are pried apart. They are being displaced from each other when the compound binds. So the compound acts as a wedge. It goes in between these two subunits and acts as a wedge, pushing these two subunits apart. So this is how far we are with these ADAPs. They are in the half a micromolar IC50 value. And we wanted to push things to nanomolar. If you want to go into the clinic, usually you have to be in the nanomolar range. So this meant that we are still working on these, trying to optimize them further to see if we can push them further, but that's work in progress. At the same time, we started looking at other chemical scaffolds that could do the same trick. And these are the TR compounds. So these compounds look something like this. For those of you who are into pharmacology or medicinal chemistry, this moiety is called imipridone. So it's imipridone-based compounds. And the way we ended up working on these is actually quite by accident. So there's this guy, which is called ONC201. So it's a compound called ONC201. It was actually already in the clinic. And it was thought actually to target this dopamine receptor, D2. And it was found to actually have very nice inhibitory effects on multiple cancer cell lines. And there were 15 clinical trials going on with this compound. And people were wrong. It did not target the dopamine receptor. It actually targets ClpP. So we collaborated with this company, Madera Therapeutics. It's a company in North Carolina in the States. And we generated a whole big library of these compounds, and we call them TR compounds. And I'm just going to show you the 5 that we concentrated on. So they are numbered TR 27, 57, 65, 107, and 133. And they are basically different analogs of the original ONC201. So initially, we checked these compounds against 60 different cell lines, and just to see their effect on cells. And we can see growth inhibition. So this is showing you TR57 and 107. TR57 is in blue, and the 107 is in red. So we have growth inhibition for all these different cell lines, or even for a couple of them, we have cell killing as well. Then we did the same experiments that I was showing you to you before, whereby we checked the IC50s against MB231s. And the MB231 are breast cancer cell lines. And what we see that the IC50s now are in the nanomolar range, like 90 nanomolar, 9 nanomolar. So they are really good compounds in the nanomolar range rather than micromolar. The original ONC201 is in the micromolar range, 1.7 micromolar. If we get rid of the ClpP, we generate a cell line, an MB231 cell line deleted of the ClpP. What we see is that these compounds don't work anymore. So they don't hit anymore. So again, we are on target. The same set of experiments were done on the HEC293 cells. Again, we are in the nanomolar range. And the KD on the protein is around, again, 20, 30 nanomolar. So they are really much more better compounds than the ADAPs I was describing to you before. So the medicinal chemistry approach, for those who are interested, initially we worked on the imipridone moiety, and then we worked on the eastern and the western fragments. So this is how we would do it. So on the eastern fragment, we found that we need something that's either an alkyne or a cyanogroup, and this did the trick. For the imipridone moiety itself, we can actually reduce the complication of that moiety by having maybe something like a pyrimidine. So much more simpler. So we're trying to simplify the chemistry. So this is much more simpler. And this one is a pyrimidine dione, and both work very nicely. And finally, on the western fragment, we see that we need a chlorine or bromine, something that's a bit electronegative. And this did the trick, and that's why we are in the nanomolar range in terms of these compounds. So subsequently, we saw the crystal structure of the compounds with the ClpP, all 5 compounds. And again, they bound in the same site at this very hydrophobic pocket. And we have 7 on one ring and then 7 on the other. So 14 compounds bound to that ClpP ring. Okay. Freezing again. So I should have simplified this. Okay. So we subsequently solved the crystal structure of these compounds with the clip P. And if you just concentrate on this one here, so you can see this binding pocket, and the compound fits extremely nicely. So it has great shape and surface charge complementarity with these hydrophobic pockets. The imipridone moiety itself sits flat. And then there are one sub-pocket here and another sub-pocket there, where the eastern and the western fragments are sitting beautifully, and very nice charge and shape complement with very nice charge and shape complementarity. Okay. Yeah. So this is another view. So this is a top view, and you can see the imipridone moiety on the top, and then one fragment going down this way, the other fragment going down the other way. And if you look at the pocket itself, you can see that there's maybe extra space down here. And this is something we are considering to build further these compounds to increase their affinity. But we are quite good. We are in the nanomolar range, and the company is quite happy. It's something that they are going to take into the clinic. So I want to show you next is the comparison between the ADAP and these TR compounds, how they sit. I think we're going through structures, so that's why it's having a hard time with it. When we tested it, it wasn't that bad. Okay, yeah. So in purple is the ADAP compound, and in the orange is the TR compound. And as you can see, the ADAP is much bigger. It has stuff going out of that pocket, extra things. But on the eastern fragment, it fills, both the ADAP and the TR compound fill that space nicely, so there's no problem. However, the problem is actually on the western end, whereby you have the orange TR compound fills that space very nicely, but the ADAP has this alkyl chain that does not fill that space very nicely. And that's why we are in the 0.5 micromolar range for the ADAP, while for the TR compound, we are more in the nanomolar range. So this is where we are with these compounds. And wait again. So let's see. Okay, sorry about all these interruptions. Actually, if you want to ask me questions, since we have time as things restart. Yeah.
How you calculate this biochemical concentration to in vivo concentration? So what is the kind of algorithm used for this purpose?
Yeah, so basically we can, for biochemically, we can measure binding constants using ITC or SPR, like biophysical methods, we can do that, or by activity assays. For cell-based assays, we can do just titrations and then measure cell viability at different concentrations, and gives us a titration curve, and therefore that gives us the midpoint of that titration curve with the IC50.
Extrapolation.
The extrapolation
from biochemical cellular assays to in vivo.
Yeah, to in vivo, exactly. So basically, this is the flow that we've been following, whereby we need something that's nanomolar on the protein level with the biophysical assays, nanomolar on the cell level with the IC50 assays, and then we take it to mice. So in mice, then we have to follow a certain dosage procedure. Initially, we start very high in dosing to see toxicity as well as to see effect on the cancer. So this is something actually I'm planning to show you as well.
Professor Waleed,
Yes.
My name is Dr. Abdurshid. Basically, I'm a regulator. Now is a part of comstech.
Good to meet you.
My question is Department of Biochemistry or Department of Chemistry, University of Canada, they have conducted phase 0 clinical trials, phase 1 or phase 2 or phase 3?
Yeah, so actually for the TR compounds, this is being done with the company in the US. So those trials are being actually conducted in the US, not in Canada.
Thank you.
Okay, Assalamu alaikum.
Alaikum salam.
Doctor, it's really a pleasure listening to you. This is
Yeah.
Dr. Pakiza. And I was just wondering, okay, like, can you tailor the compound according to the space available? Like in silico tailoring can be done to fit the compound more like nicely to the space available?
Exactly. So this is what we are doing. We are doing structure-based drug design. So as we solve the structures of these compounds, then we can redesign them to try to get higher affinities. It doesn't always work as we think. So we basically we have X-ray structures that are being solved. We have the compound, then we see that there's an extra space. We try to occupy that space. Typically, this is how we've been following that up. So we try to occupy that space to ensure that we fill all the space that can be available. However, when we go in mice, then there are other considerations, which is the stability of the compound and whether the compound can survive degradation in the bloodstream or the liver. So there's other considerations when it comes to the pharmacology aspect of it. And that's why from the medicinal chemistry aspect of it, the chemists want us to reach nanomolar, or the company, I should say, want us to reach nanomolar before they talk to any investors. Let's put it, I mean, money always talks. So they want to see a nanomolar compound before they talk to any investors. And then from the investor point of view, they say, okay, this seems to be a good compound. We have to show specificity that it is not targeting anything else. Because if you don't show specificity, again, they don't like that either. So we have to show specificity, we show efficacy in mice, and then we can push it further into a clinical trial.
Telling us about the entry of EDIP into the cell, like how you manage it. You said that it is a quite big compound, and there had been, I missed that point. Sorry.
Yeah, so basically when we, if you look at the ADAP, usually for compounds to enter the mitochondria, they have to have some positive charge character to them. So like if you use mitochondrial staining, usually like a mitotracker for those who do cell biology, that's a very positively charged compound. That's why it lights up the mitochondria. So usually you want something that's has a positive character to it. And also something small, because I did not think that these ADAPs would cross the double membrane of the mitochondria. They might cross the plasma membrane, but I did not think that they would cross the double membrane of the mitochondria. Nevertheless, I was proven wrong. Somehow nature does not always agree with the way we think. So it did work. And why they worked, I am not sure, to be honest. Looking at the structure, I would say that compound should not go into the mitochondria, but all evidence we have so far, it is going into the mitochondria. So what we are doing now to try to push these compounds, and that's why we are stuck at a micromolar range with them. I think there's a portion is going into the mitochondria, but the other portion is still stuck in the cytoplasm. So what we are doing now is trying to introduce these positively charged moieties on the ADAPs to try to see whether we have better entry into the mitochondria, and therefore maybe we can push them into nanomolar range.
Assalamu alaikum.
Alexa?
This is Dr. Anam. I have a question that how you select the particular protein for the compound, like the pockets you have mentioned and the active sites, and how it can relate with the cytotoxicity result. You show the binding constants or binding and the concentration of the particular compound in the graph. So I have a question that how can you correlate with the particular protein binding sites and the binding constants? That is my question.
So you're asking me how I correlate
The cytotoxicity
cytotoxicity
with the binding constants.
Yeah, so actually I showed you the correlation. So basically we have a binding constants that we measure at the protein level. So basically we're doing binding constants of compound of the ADAP, say, with the ClipP protein. we get a certain binding constant. So you can do ITC or SPR or activity assays, and you get a binding constant of the compound to the protein, to the purified protein. And then we have IC50s on cells. And we saw that we generated a whole series of compounds, and we see that the binding constants that we get against the purified protein correlates very nicely with the IC50, which tells us then that these compounds are on target, that they are targeting ClpP in cells. Because if they are targeting something else, we should not see that correlation.
So one, the basic question is that how can you select? There are lots of active protein sites for a particular, just like you say the cancer, lung cancer or the other cancer. So how can you select the particular active site for the target drug?
Yeah, so basically in this case, we are not selecting the active site. So we're not going after this catalytic triad, the Searhis-Ask catalytic triad. We're not going after the catalytic triad of the ClpP. We're targeting the docking site of the cap. So this is where the ClpX docks. These hydrophobic pockets is where the cap docks. So we're going after the docking site because we want to basically activate the ClpP. So there might be other sites, cryptic sites. And actually, there's a compound that has been described by another group, which seems to bind somewhere else, although their data is a bit iffy. But there are cryptic sites that you can go after as well. And I mean, that's something we are also interested in. If there are other sites we can use to play around with the activity of this protease.
Thank you.
Okay, we're good to go. Okay, let's see. So I think this slide is done. Yeah. So just to show you the mouse experiments. So here, these are xenograft mouse models, whereby we took MB231 cells and xenographed them on mice. And we then used either vehicle control, or we dosed the mice with a certain periodicity. And what we can see is that the tumor volume after 26 days is reduced compared to the vehicle control. So this is one experiment. The other one is that we check the viability of the mice. So in black is the control group, and the mice die early on. But in red and blue are how long the mice survived when they were dosed with this TR107 compound. And finally, the other thing I want to show you is that we can measure mitochondrial DNA, because we are targeting the mitochondria, right? So, and if we are targeting the mitochondrial DNA should be reduced. And this is what we actually see, that the mitochondrial DNA levels in the dosed mice is much lower than in the control group. So these are the mice experiments telling us that the compounds are working in a mouse model. So this then takes me to the next set of experiments, which basically, what are the consequences of ClpP activation on the cell and on the protein? So this is, again, going back to the basic principles, trying to understand when we dysregulate ClpP, what does it cleave? Because, so what are the proteins that are being targeted by a dysregulated protease? So we did the following experiment. It's a mass spectrometry-based experiment called Titane, whereby we grow the cells, these MB231 breast cancer cells, we grow them and then divide them into two. One group will be treated by a drug, by the TR drug, while the other group is treated by the MSO. So subsequently, what we did is the following. So this is a description of the Titane analysis. It's a bit more involved, but what we are trying to get is what are the N-termini that are being generated when we cleave with the activated ClpP. So the activated ClpP is targeting now many proteins inside the cell, right? And we want to know what these proteins are and where the ClpP is cleaving. So we did this experiment, and the purpose of this experiment is to know the new N-termini that are being generated, because if the ClpP cuts, so I'm asking what is that sequence that's being cut? And the way this is done is briefly, again, you can ask me about it later on if you want more details, but basically we take the cell lysate, we unfold the cell lysate in guanidine, and then we block any free amino group with by dimethylation with formaldehyde. So any free amino group will be blocked by formaldehyde, and this is shown here by these yellow squares, yellow diamonds. So all the amino groups will be blocked, lysines, as well as the N-terminal amine. So everything is blocked. Subsequently, we digest with trypsin. If you digest with trypsin, then you're going to generate these new amino groups, right? Then you react these guys with a very hydrophobic tag. So these will be reacted by a hydrophobic tag, and this is shown schematically in red. So all these fragments will have this hydrophobic tail to them, while this fragment will not. So I'm trying to get the sequence of this fragment, right? So subsequently, I can take this and run it on a C18 column and a reverse phase column. And then these hydrophobically tagged peptides will be removed. And this peptide will be the one remaining. And then I can take this peptide, do mass spec on it, and identify what that is. So this is called the high-chain approach. And it lets me identify the N-termini of proteins in a cell lysate in a high throughput manner. So it's really quite powerful. And this is what we did. So what we see, we identified around 200 proteins that had these new N-termini. And we found proteins that are in the mitochondria, but also in the cytoplasm and the nucleus that were being cleaved by an activated ClipP. So now you can ask me the question, how come you can cleave a protein in the cytoplasm and while ClipP is in the mitochondria? And I don't have a clean answer for you. I can wave my hands and say, I think what is happening is sometimes the mitochondria is leaky. Some of the ClipP might end up outside of the mitochondria, whereby you are then it's being the activated ClipP is cleaving cytoplasmic proteins. But what we see in general is that any mitochondrial protein that is getting cleaved is actually its levels go down, while any cytoplasmic or nuclear protein that's getting cleaved, its levels go up. So somehow it's getting cleaved but stabilized if it's cytoplasmic or nuclear, but if it's in the mitochondria, it gets cleaved and it's destabilized and it's degraded. So since we now have the sequences of these proteins, we know what they are. What we can do is check whether there's any consensus sequence that's being targeted by ClipP. So is there a consensus sequence that's targeted by this protease when it's activated? And the answer that seems to be no. We don't find a consensus sequence being targeted. But what we find is that ClipP likes to cleave in coiled regions. And that's something you might have expected, but now we have proof for. Basically, any flexible loop will be cleaved by the ClipP protease, by the activated ClipP protease. So this is quite fantastic because now we have an idea what ClipP is going after. So once it's activated, it seems to go after any flexible loop and goes and clips it. So this is, if it does that, then the levels of these proteins go down. We did find ClipP to cleave at helices and sometimes strands, but those are minor and they tend to be stabilized. So they are stable if this happens. And I'm showing you here some examples of these proteins that are being cleaved by the ClipP. And this is, so ClipX itself, the cap itself is actually being cleaved by the ClipP and it's disappearing. SDHA is part of the complex 2. If you're familiar with mitochondria, this is the succinate dehydrogenase. TFAM is the major transcription factor in the mitochondria, and that's being cleaved. And there's a few others. I'm just going to highlight. This is the RNA polymerase is also getting cleaved. And finally, the chaperone itself is getting cleaved. This is HSP60 chaperone is getting cleaved as well. So now we have an idea of what this activated ClipP is targeting, and we have an idea of what sequences it likes. It doesn't really like a specific sequence. It just likes to have floppy bits that it can go after and cleave. So what's happening on the protein level, without going into too much of the biochemistry, but what's happening is the following. As I mentioned before, it seems if you look at the ClipP in the apostate, in the absence of any compound, and you look at the entrance pore, so this is the entrance pore, there are these axial loops that are quite floppy and they block the entrance pore. And there's this ionic interaction network at the entrance pore that is formed. And that's what keeps the entrance pores narrow, small and narrow. But when the compound comes in green, this green compound then acts as a wedge between two subunits. So it's inserting itself between two subunits, and that breaks this network of interactions. And as a result, the axial loops become ordered and they are retracted, and the entrance pore becomes much bigger. We also can see that there's a lot of dynamics in this protein, and we can measure that dynamics using different approaches. This is one experiment that I'm showing you. This is by NMR. In this NMR experiment, we add a label either at the axial loop or in the handle region here, which I mentioned to you before. And this label is C13 labeled. So it's C13, and therefore we can monitor what's happening to that C13 label. And what we see is that in the apo state, in the absence of any compound, in the axial loop, this C13 label has many peaks, meaning that it's basically moving a lot. So it's sampling many different conformations. But then when I add the compound, there's only 2 peaks. Basically, it's being stabilized in one of 2 states. And then the axial loop is actually being stabilized. And the same thing happens in the handle region. In the handle region, what we see is that we have at least 2 states that we can see. There are 2 peaks in that region. So there are 2 states. But when we add the compound, the ADAP compound, we switch it to 1 state. There's only 1 state that's being stabilized. So again, meaning that we are stabilizing the dynamics of the protein when the compound is added. So to summarize these observations, as well as a lot of our biochemistry, what we notice is that in the apo state, ClIP is actually quite a flexible protein. There's a lot of flexibility in the red parts here, in the axial loop regions, as well as in the equatorial part in the yellow regions here. So it's a very flexible cylinder, if you will. There's a lot of flexibility. But when we add the compounds, this flexibility is lost and becomes a much more rigid molecule. And this is there's rigidification and in the axial loops, and there's also rigidification in these equatorial regions. So if you look here at the top view, so the axial loops are quite flexible. So they act as basically, we call them entropic barriers. So they are barriers to the entrance of proteins into this cylinder. But then when you add the compound, then these loops are stabilized and they open up and allow things to go in. And this is how ClIP activation works. And this is shown schematically here, whereby you have opening of the entrance pore, and there's actually tightening of the belt. So you're basically eating less, or actually ClIP has to eat more, but somehow it becomes thinner as it eats more. So I think this brings me to the end of my presentation. Sorry again about all these interruptions, but basically, I hope to have shown you that this regulation of mitochondrial ClIP is a new approach to develop new anti-cancer agents. So if you think of it, most anti-cancer agents are inhibitors of something, right? You want to inhibit some pathway, inhibit some enzyme. But in our case, we're activating. That's something completely new. And we're the first ones to propose it. And this activation leads to very interesting biochemistry. So I want to thank my group. So this is the group back in July. The people who led this effort have been, actually Mark has been really instrumental. He's a crystallographer. Keith is the cell biologist. And Mariam in the back here, she's the one who did a lot of the biochemistry. So with this, I will stop and take any other questions. Thank you.
Thank you very much, Dr. Waleed, for your very interesting and insightful presentation. So we have the next few minutes for question answer session. So if anybody from the audience has any questions, you may ask now. And also our online participants can type their questions in the chat box.
Okay, so thank you very much for this very enlightening and informative talk. Yeah, sure. So I have a few questions. First of all, I want to ask that, did you also use MB231 cells for the ADAP experiments? Because you showed that it's HEC293 cells. Did you also check it for MB231 in there?
Yes, we did that also for these cells, the MB231s, for the ADAPs. Yeah, we've done many different cell types for the ADAPs as well.
Okay, was it working with that too? Because it's like breast cancer cell line, and so that would actually be very important for the breast cancer patients as well.
Yes, so they are working as well. And actually, currently, we are collaborating with the group at the NIH. We're testing them on mice.
Okay.
the ADAPs. We're doing the mice experiments right now.
Okay, and I was also wondering about this apoptosis thing that it triggers, and you have shown nicely, but I have a few more questions. The proteins it degrades, what do you think, which protein degradation actually triggers the apoptosis process?
So we think it's actually not necessarily just one protein. One thing I did not highlight, but we saw, for example, MCL1 is being degraded. And as you might know, MCL1 is an anti-apoptotic protein. So basically, you're getting rid of an anti-apoptotic protein and you're inducing the caspase pathway. And so,
Yeah,
but we don't think it's a single protein. It's a group of proteins that are being degraded that eventually causes that.
Because I thought it's really interesting to know, like, how does it trigger the intrinsic pathway, like which BCL2 related proteins?
Yeah, exactly. So MCL1 is one of the BCL2 related ones.
Okay.
And it's anti-apoptotic. So basically what's happening is that you're getting rid of that. So CLIP-E, the activated CLIP-E is getting rid of that. And eventually apoptosis is induced.
Thank you very much. And I have one more question.
Sure,
if I may
yeah,
ask.
go ahead.
How will you make sure that these compounds, they will specifically target the cancer cells and not the normal cells of the body? So
Yeah,
how
very
can
good
we-
question. And I think we are saved by the fact that the CLIP-E levels are higher in the cancer cells. So this is something I always get because CLIP-E is everywhere, right? So and we are always worried about toxicity. But for some reason they work and they work very nicely. And I think we are saved by the fact that the CLIP-E levels are much higher in the cancer cells compared to the normal cells. And that's why we really need to push to nanomolar. So we reduce any toxicity.
Thank you very much. That's very great.
So there are questions in the chat.
Yes,
Do you
after
wanna-
this,
Oh, there's one more here. Yeah.
Thank you so much for your talk. I'm Dr. Shahrukh from NAST. I'm not a conventional biochemist, but a technologist. So I'm not sure whether my questions will be very much relevant to your talk. I have one question that if can the CLIP be dysregulate using if the dysregulation is based on the charges and complementary, can we use any nanoparticle to do the same for dysregulation?
So you want to use nanoparticle to dysregulate the protease?
Yeah, instead of using the peptide which you made, the protein,
compounds?
Yeah, the compounds.
But nanoparticles are huge compared to these compounds. I'm not sure how.
Yeah, that's why I wanted to ask what is the size of this CLIP-E protein on the mitochondrial membrane?
No, the CLIP-E itself is around, say, 120 angstroms by 120 angstroms. So this is the size of the cylinder. So they are very small. I mean, I think nanoparticles are much bigger.
Okay,
Yeah.
and the second question is that can your compound do the same activation and deactivation to the the cellular receptors as well, the same way? Because usually we do inactivate the cellular receptor using the anticancer drug. But can the same dysregulation approach be used for the cellular receptor as well?
Yeah, I mean, you can think of many. So this is something new. So this regulation is something that we proposed. And now there's a term for it, which is called paracatalysis, whereby you are forcing an enzyme to do something it does not typically do. So it's called paracatalysis.
Mhm.
And in theory, you can do that if you can find the right compound, you can do that even for these receptors that you're mentioning.
Okay.
And so there's other dysregulators that have been described for other proteins as well.
Okay.
But the mechanisms are very different.
And if I understand correctly, so it's of course the mitochondrial protein, right? So it's the mitochondrial dysregulation, right?
Right.
So it means that your compound first has to cross the cell membrane and then go into the mitochondria.
That's right.
So it is. Can you make this? Can we make a dimeric protein or dimeric, a composite compound so that it can only, if we cannot make it more specified to the CLIP-E of only cancer cells, can we combine or can we make a composite so that it can be only going through the cell receptors of the cancer cells there have been, because the cellular receptors of the cancer cells are in much more, researched upon for a very long time. So can we have any?
Yeah, so that's something we have not done, but I see what you're trying to go after. Basically, you want to fuse our compound with another compound that targets the
Yes.
cancer cell receptor.
Yes.
Yeah, we have not done something like that. It might be easier to go and conjugate the compound with an antibody that targets the cell receptors, actually. So that has been done. But fusing two compounds, that has not been done. But fusing the compound with an antibody.
Or a nanoparticle receptor.
Oh, okay. You like your nanoparticles, for sure. If you can make the nanoparticles specific to your cancer cells, yes.
Thank you.
Yeah.
Okay, so we'll take the last question from the audience, please. Thank you.
Sir, I'm Neza Fatma. Basically, I have a question. Do we need to fill all the 7 pockets of the protein, Cleaving protein, to make it non-specific, or the partial filling would be enough? What was the effect of partial filling of the pockets on the cell?
Yeah, so this is a very good question, and that's something actually we studied in a lot of detail, because it's something that we were worried about. How many of those sites we need to fill, right? So you have 14 sites in the cylinder, and we want to make sure, do we need to fill all 14 or just 1 is enough? So actually it turns out if you can fill 1 per ring, so 2 for the tetradecamer, then we would be okay. So all we need is basically 1 site, and that would be enough to start the activation process. It might not be as good as filling all 7 or all 14, but at least 1 is enough.
If 1 is enough, then it means its concentration can also reduce more. We can go to the more lower concentration.
Sorry, I don't think your mic is working, but can you repeat the question?
Sir, I'm saying that if only 1 pocket is enough, if the filling of 1 pocket is enough, then its concentration would be more lower. We can reduce its concentration.
Right. So you can reduce the concentration, and this is what's important for us, is that we don't want to go to too high concentrations to reduce toxicity or cross reactions to other things. Yeah.
Okay, sir. Thank you.
Yeah.
All right, thank you very much. This brings us to the conclusion of this event. Request Dr. Iqbal to kindly present the Shield of Honor to Dr. Waleed.
Thank you.
Thank you very much, everyone, for coming. Thank you for your time. Please join us outside for a group photo and then high tea in the lobby area. Thank you.